Welcome to our review on monoclonal antibodies. So the first thing we're going to look at is what is a monoclonal antibody? And quite simply, these are monoclonal antibodies because they're produced by a single clone of cells. Now what that actually means is we're going to produce these in the lab by using a type of cell called a hybridoma. A hybridoma is a fusion of two types of cell, myelomas, which are cancer cells, and lymphocytes, which are white blood cells that produce antibodies. So once we've fused those two types of cells together, we've got a hybridoma, and they then produce these antibodies for us. What we do is we actually design these monoclonal antibodies to target a specific type of cell. And the way we do that is that they're designed to bind to antigens of our target cell and either kill it or prevent it from operating effectively, depending on the end result we're aiming for. So if we think back to what we looked at in terms of our white blood cells, we know that lymphocytes produce an antibody which is specific to a particular antigen. So we're just using that feature of our lymphocytes to then give us something that's able to target a specific type of cell. The next thing we really need to consider is how we produce them. So we're going to start off with some good old genetically modified mice. So we inject those GM mice with the required antigen, and the body of the mouse will then produce antibodies using its lymphocytes to that specific antigen. We then collect the lymphocytes, and then we fuse those lymphocytes with our myeloma or cancer cells to make the hybridoma. So what we have to do is carry out that fusion stage because the lymphocytes can't actually survive outside the body on their own. So if we didn't fuse them to the myelomas, they wouldn't last. And the reason that we use myeloma cells is that they can reproduce indefinitely. So that means that we can keep producing more and more of them for an indefinite period of time. As our hybridoma cells then reproduce, they form clones. And remember, a clone is an identical copy. So that means that each clone will produce the required antibody. And we can then harvest all of those antibodies that have been produced by these hybridoma cells. And those are the monoclonal antibodies that we can then use in a range of different things. In terms of why we actually want these monoclonal antibodies then, we've got a range of different things we can use them for. One of the more common ones that you've potentially heard of are in pregnancy tests. So those little sticks that someone pees on and then you get a little blue cross or line if you're pregnant, then that uses these monoclonal antibodies. So what I've given you in the top right is a little diagram of how it's actually set up. So the bit that the person actually pees on, then you've got antibody stuck onto a little blue bead. And then a bit further on in the window that you're going to look at, there's antibodies that are actually stuck down to the actual surface there. So when someone actually urinates on that stick, what happens is if they've got the HCG present, then it's going to bind to the antibody on the end of the little blue beads. As the ill moves up the actual strip, it reaches the window that we look in, and because there are antibodies stuck down in there, then the HCG will bind to those stuck down antibodies as well, and we end up with a blue line as all the little blue beads line up and are then stopped from moving. They're immobilized. If obviously the lady isn't pregnant, then what happens is those blue beads won't stick to those stuck down antibodies and they move past the window, so no blue line. Obviously, when people are using these home pregnancy tests, then they do want to check that the test is actually working. So they will also, in some cases, have a control window. Now, the whole idea behind that is to check that the test has actually worked and that those little blue beads have moved. So what happens is that control window contains antibodies to the antibodies attached to the blue beads. So it's like an antibody of an antibody. So that as those little blue beads move and they go past the window that obviously has the antibodies to HCG, doesn't have HCG, won't bind there, so it keeps going. As it reaches that second window, the control window, then the antibodies on the surface of the blue bead are going to bind to the antibodies that are stuck down there. And as a result, we get a blue line in the control window. A second way we can actually use monoclonal antibodies is in treating diseases. When we consider different cells, then they've actually got different antigens on their surface. So cancer cells, for example, have a special kind of antigen on the surface called a tumour marker, which means that if we want to target cancer cells, if we use a monoclonal antibody that's going to bind to those tumour markers, then we can target drugs to just the cancer cells. So what we do is we attach the cancer cell onto our antibody, and then the antibody is going to attach to the tumour marker, delivering the cancer drug to the cancerous cells. A third way that we can use our monoclonal antibodies is when we're actually looking for certain chemicals. So in the lab, we may be looking for hormones or certain banned chemicals, for example, in the Olympics, and we can do these tests using a monoclonal antibody. So a good example of this is the ELISA test, which tests for those banned drugs in athletes' urine during events like the Olympics. We can also use them to identify the different strains of gonorrhea that may be present in a sample. So it just allows us to identify certain chemicals or hormones that are present, and not only that, but we can also measure the level of them. The final way that we need to know that monoclonal antibodies are used is in locating specific molecules. So, for example, if we wanted to locate cancerous cells, then what we'd do is we'd make a monoclonal antibody that's specific to the molecule we're looking for, and we'd attach a fluorescent dye molecule to the end of it. When we then mix those monoclonal antibodies with the thing we're studying, what happens is they will bind to the antigens on the surface, as the antibodies are designed to do, and then that dye will fluoresce or glow. And you can see the image on the right there that's showing skin cancer cells that are literally glowing because we've attached monoclonal antibodies with that fluorescent dye on there. Hopefully at the end of this video you can describe what monoclonal antibodies are and how we actually make them. You can also recall some of the uses of monoclonal antibodies and then explain how we use them in each of those examples.